Alright, this is the 10-minute wrap-up of Chapter 12, Mitosis in the Cell Cycle. We will be talking about the cell cycle, mitosis, and cancer. Basically, we have cell growth kind of going on here. What's going on here? We have the cell theory in that all living things are made up of cells. So everything has to have a cells. What does cell division do for you? Cell division replaces lost or damaged cells. It's involved in reproduction and growth. Asexual reproduction is where an organism reproduces without the fusion of egg and sperm. You're making clones of one another. During sexual reproduction, you are requiring a fusion of two different gametes, egg and sperm. Meiosis is the production of egg and sperm. And so what this figure is showing down here is that mitosis is involved in reproduction, two-cell dividing, growth and development, so when we go from one cell to a multicellular organism, as well as tissue renewal. Again, you need to know what a chromosome looks like, what these sister chromatids are. These sister chromatids are copies of the DNA of one another. They are held together by a centromere. This chromosomal arm is also a chromosome. And then again, when these sister chromatids separate, they will produce two different chromosomes, and again, they will be exactly to one another. You should know what a cell cycle is. Again, interphase is where the cell spends most of its time and what it's doing. There is a G1 gap 1 phase, S synthesis phase, and G2 gap 2 phase, and then our mitotic phase, which is a lot of times only about 10% of it. Your lecture here tells you to think of it as a clock. I think of it as more of a signal on a washing machine. Not that I've done very much washing in my lifetime. You should know what's going on during interphase. Our chromatin is there during G2 phase. It is replicated during prophase. These chromosomes condense and come together, and you can start seeing the structures on them. During metaphase, the sister chromatids will line up on the axis of the cell, and then during anaphase they are being pulled apart, and you can see the daughter chromosomes that are there. And then during telophase you will see actually this the cleavage furrow and the beginning of the two new cells coming up. Again, here are some really interesting photographs of being able to see this, and I would take a look at those a little bit later. And again, mitonic spindle at metaphase that we see here in this aster. We see that the sister chromatids, their centromeres, are attached in the structure that we call the kinetophores, the movement that we see of here. And we will talk a little bit later of how those kinetophores work. In the evolution of mitosis, prokaryotes evolved before eukaryotes, and mitosis probably evolved from what we call binary fusion, which is basically we make a structure that is attached to the opposite ends of the cells, and we are able to pull those structures apart. Cytokinesis in animals and plant cells is a little bit different. In animal cells, we form a cleavage furrow, which is basically a contractile ring of microfilaments that successfully squeeze and separate the cells, whereas in plant cells we see vesicles forming the cell plate. The cell plate produces a new cell wall, and then these cells can basically fill up with fluid and extend. How does cancer relate with mitosis? A number of general ideas. Cancer is uncontrolled cell division. Cancer cells have genetic damage throughout them. Proto-oncogenes are normal genes that make normal proteins that promote cell growth, and they are what they are before they turn into cancer. Tumor suppressor genes are normal genes that make normal proteins that inhibit growth. At least 2 mutation events, and usually more, are required for a cell to become cancerous. So typically whenever we've looked at a cancer cell before, if it is truly cancerous, that there's a number of different things that are wrong with that cell. Mutations can be hereditary, roughly about 10%, or can be caused by environmental factors, which we think is the vast majority of cancers. And again, this is due to things that are carcinogens, which do damage or mutate the DNA. Anything that does that can do it, from UV sunlight to movement. Again, if this is an idea here of looking at genetic causes of cancer, if your friend has a genetic trait that will cause a cancer, then again, it does not necessarily mean that they will get breast cancer later in life. But hopefully you will be able to talk to them about that. Again, why is it important that cell division happens at the appropriate time? This is looking at breast cancer that's inside there. Again, this a tumor can spread to other parts of the body, malignant tumors, when it goes on. Basically, cells have signals to tell it when to start dividing and when to stop dividing. And again, there's a number of different mutations that can go on. So if you normally have a gene that's responsible for stopping the growth of a particular cell, and you knock that out, then the cell will grow exponentially. If you normally have a gene that's responsible for turning it on, and you get excess of it. And so what this is basically showing you here is that we're looking with an organism with 4 chromosomes, and that in order to get a mutation, a malignant cell, that a number of different mutations have to occur. The cells are very tightly regulated as in terms of their growth, and we tend to not see a mutation, malignant growth, unless something is happening. Again, know all these terms: mutation, proto-oncogene, oncogene, tumor, apoptosis. Again, how is cancer treated? The best way is to basically cut it out and get rid of it, but again, we can't cut out all big important parts of the body. Typically, we will burn and we will poison the cancer out. Ideally, we are killing the cancer cells and not your cells. Again, we think about cancer like a biologist. Again, in the cell cycle, what we're looking at is cell division, producing 2 identical cells. We looked a little bit at cancer. Again, plants are a little bit different in that they're forming a cell plate instead of the furrow. Be able to look at this, look at the cell cycle, be able to identify parts of the cell cycle. And evidence for cytoplasmic signals are basically where we took cells that are synchronized in certain levels, and we would put them together. And so when we put an M and a G cell, that we would automatically go to M. When we put together an S and a G1 cell, that we would go to the S phase. And so there is something within the cytoplasm that controls this. And again, it's a lot like a clock or another structure, another thing. Again, we're looking at cancer cells tend to not inhibit their growth, and so what they do is they will grow on top of one another. And basically what we see here is, let me get this, cancer cells. So normal cells would exhibit what we call density-dependent inhibition, and cancer cells will not. They will kind of move on top of one another. And again, that's basically it. Thank you.